Thanks, Ahmed, for the introduction. Good afternoon, everyone. My name is Yasmine Gomaa, and I'm a senior scientist and the associate director of the Laboratory of Drug Delivery. Of course, thank you very much for the invitation to be among this year round of speakers for the conference. As the title implies, today I'm going to speak about microneedle-based drug and vaccine delivery. And before digging into specific examples of drug and vaccine delivery, let's go over just a brief overview of what are the microneedles, how they work, why they are so special, and things that would help us understand more the role in drug and vaccine delivery. So microneedles have been developed initially to allow transdermal drug delivery by just breaching the superficial layers, which is the stratum corneum, and that create microchannels in the skin that would allow the passages of drugs. And because of the size of these channels that are in the micron range, that would allow bigger molecules like macromolecules, as well as drug delivery systems such as nanoparticles and microparticles, which is not the case with a conventional drug delivery, conventional transdermal drug delivery, where we are confined with certain physical chemical characteristics when it comes to molecular weight, PK or partition coefficient, log P, and all these properties of the drugs. Here on the top right, you would see where the microneedles would be located once inserted in the skin, and this is just in comparison with the other injection routes like IM, subcutaneous, and the intradermal injection. And on the left here, you will see how big it is in comparison with 1 cent. So in terms of geometries, they are broadly classified at the beginning into either an in-plane or an out-plane. So an in-plane, it would be, the needles would be just in the same plane of the substrate. So they are basically a planar form. They are very easy to manufacture. You can control the length, and they are very much suitable for microsensors and micropumps. However, if you want to go with an array in a two-dimensional structure, it's the out-plane would be better in this case. So as you can see here on the top row, they come in different shapes. Here, this is the conical, pyramidal, star-shaped, and this is very unique shape, kind of resemble how the hypodermic needle looks like. Also, if we look at the shaft of the needle itself, and these are here stainless steel microneedles, you can see that they can be just as solid as in here, or have more complex shape or structures where these pores can be actually filled with drug solution and then be delivered into the skin. In terms of length, they can vary from 100 to 1,000. You can surely go beyond that, but then you will have an issue with having a painful delivery, which is something that we don't want to do. And we always advocate microneedles delivery as being pain-free. Also, they have different basal diameter, range from 130 to 300, and different aspect ratio, different tip angle. They are often also arranged in the form of an array of hundreds of needles, and they can range in like size from 1 centimeter squared to up to 10 centimeters squared. Generally, the approaches of transdermal drug delivery using microneedles are divided into 4 approaches. The first approach is termed the poke and patch, which is basically when you use a microneedle patch that is made of a solid material. Once you insert it in the skin, it would create a pores. Once you remove this patch, these pores will act as a conduit through which the drug that you would apply afterwards pass through the skin. So it's like a 2-process step. The second approach is called the coat and poke, and this would involve using coated microneedle, in which the drug solution or the vaccine is being coated around usually a non-dissolving needles, such as made of metals, for instance. And once inserted in the skin, this would start to dissolve and the drug is being released into the skin. So it's, again, a kind of a 2-step process where you would have to insert and then remove the needles because they're not going to dissolve. The third approach, which is the most common nowadays, is the poke and release, or what we call dissolving microneedles. So these basically are made from dissolving excipients, polymers, and sugars, and some stabilizing excipients that we add to the casting solution, mixed with the active that you want to deliver. So once inserted in the skin, these needles start to dissolve and deliver the drug. So it's a one-step process, and that's why it's more appealing. And most of the research nowadays on microneedle drug delivery is based on using dissolving microneedles. The final approach is the poke and flow, and this is basically uses hollow microneedles. And as you can imagine, these are only used for delivery of liquid. So we have been using this quite often in delivering drugs through the eye, like ocular delivery. Recently, there have been another type or approach of delivery using microneedle patch, which are the swellable microneedles. So they are made of like hydrogels, cross-linked hydrogels that once inserted in the skin, they start imbibing water, and then they will start to swell. And these actually needles embedded in the skin would act as like a continuous transport channels of the drug that is often located at a reservoir in the backing of the microneedle patch. So basically, these are intended for an extended release of the drug. So what is so special about these microneedles? So I would say microneedle patches meets public health needs. And how is that possible? First, with respect to patient administration. So on the right hand here, you see how the microneedles are being used. So basically, it's a size of like a Band-Aid that you can use. You would take it out of the protective cover, because these are needles. Of course, we have to maintain them like protected so that they won't break and they are still sharp. And then basically, you would apply it to your skin. You can use your wrist. You can use your forearm. And then you would keep it in place for a few minutes, and then you would just remove it and discard it. So it's very simple in application. So meaning that you can self-administer to yourself, or if it's not the case, then it can be administered with a minimally trained personnel. That means we don't need to have those very well-trained healthcare workers, such as nurses or doctors, to apply these to patients or during vaccination campaigns. And that would have a huge impact on the cost of the vaccination campaigns, because it's not only the cost of the vaccine, it's the cost of all the logistics that are associated with it, with respect to having healthcare workers on site, as well as other things that we're going to discuss later. One other thing is that there's no applicator involved here. It's just the pressure by your thumb. Sometimes we include a snapper or a clicker that would produce a sound, and this is just to assure the patient or the person who's using them that he has been able to successfully administer the vaccine to his skin. Also, it's a painless delivery. So I guess some of us might have a kid that he had to take to his doctor to get his vaccination shot. And I believe everyone have been through this panic attacks once they see the needle. So imagine being able to avoid all this by painless delivery, because basically you would not be able to see a needle. It's just an array of very microscopic needles that cannot be seen by the eye. So this is one of the things that is very appealing about the microneedle patches. Second, in terms of manufacturing, of course, filling vials and syringes are much more expensive compared to tablets, for instance. We expect that with the advancement in the manufacturing processes that are actually ongoing now, we can be able to compete with the price of injectables. We use very small amount of excipients given the small volume that we are working with in the microneedles, and they are very cheap compared to other excipients. For instance, here I'm showing how we make patches in our lab by a micromolding process. It's a simple process that involves having a PDMS molds where we cast our vaccine, that followed by centrifugation forces to kind of fill these needles with the first cast, which is basically the vaccine or whatever we want to deliver. And then this is followed by casting a backing or a support that can hold all these needles together, and then demolding and a drying process. So it's simple. It can be scaled up. We know that there are facilities that are actually working on making microneedles right now, but up to now, this information is confidential. So hopefully by time we can learn more about this. But again, it's a simple, scalable process. In terms of transportation and storage, what you see here on the right-hand side is a box of 50 doses of measles and rubella vaccine microneedle patches. And as you can see, it's as big as your iPhone. So it kind of uses a small space. And this is, again, very critical with respect to the price or the cost of a vaccination campaign that involves part of it, the transportation and the storage. So that would definitely have an impact on the cost of the campaign in general. This is unlike, of course, if you have an injectable vaccine where you would need the vial, you would need the syringes, and also you would need the water of injection. And all this takes space, and all this space means money. The other thing is the improved thermostability, which is a huge thing. We have demonstrated several times that with the proper formulation of our vaccines or our drugs into a microneedle patch, we can thermostabilize this. So basically, it's converting a liquid dosage form into a solid dosage form with having proper excipients that can thermostabilize it under different storage conditions of temperatures and sometimes humidity, as you would see next into the slides. So by being able to avoid the cold chain, that's $1 million of like savings. As you are aware, injectables, vaccines, they have to be stored in a cold chain that is during storage, during transportation, and at the vaccination sites. And any disruption in this cold chain means that the vaccine can be deactivated and wasted, or you would be delivering a less effective vaccine. So this is, I would say, a very big benefit of using microneedle patches. But again, this would need a lot of formulation, optimization, and sometimes it's, there's no formulation that would fit all the vaccines. You would have to tailor this formulation to each vaccine you are working with. Like attenuated vaccines are definitely different from live vaccines. So it's a process of optimizing this for each vaccine you work with. The other thing is the vaccine wastage, which again is a huge thing. There are analyses that suggest that we loses about 25% of the vaccine from a 10-dose multi-dose vial, and almost 40% if it's a lyophilized vaccine. This is a huge issue if you have a limited supply of the vaccine. Like imagine something like the COVID vaccine. If you're gonna waste that much, then this is a lot of waste. Like there are developing countries, there are low middle income settings that are in need of this vaccine that you're wasting here. So if you were be able to have a single dose that have the exact dose that you want to deliver, that can offset and get rid of all these vaccine wastage. So this is again a very good benefit of having a single dose of a microneedle patch. Finally, it's the waste disposal. Again, part of the price or part of the cost of the campaigns is the proper disposal of the needles that are generated after the injections, because you don't want to have any incidents of accidental re-stick injuries and any transfer of blood vector-borne pathogens. And that's, of course, is an issue, especially in low-income settings where there are, again, not available healthcare workers as required. In case of dissolving microneedles, once they are inserted in the skin, they start to dissolve. So basically, after 5 minutes, you don't have any needles. And that means it's impossible to reuse. Even if you imagine being able to reload this patch again with needles, that needs a lot of expertise, that needs a lot of equipment. So it's basically impossible. So there's no issue of any breeze stick injuries that would be associated with the use of microneedle patches. And of course, a reduced disposable volume. Of course, you would need to dispose it properly because, again, it might be mixed with ISF, interstitial fluids, or blood, but it's not as hassle as dealing with needles that needs to be disposed in a certain way. So in terms of application, microneedles have been applied or tested for transdermal delivery of quite a number of drugs, of immunologicals, of biopharmaceuticals, and have been proved efficient in that site, both transdermal and dermal delivery. We have done also some work in our lab on the intracellular delivery, delivering plasmids into the cells, and that was very successful as well. Recently, there have been this diagnostic application that the microneedles were able to do as well, and this is basically depending on extracting the ISF, which is the interstitial fluid, basically the fluid in between the cells and the skin. And we found that it has a number of biomarkers that are overlapping with what we could see in the plasma, as well as some unique biomarkers that can be used for the diagnosis of many diseases. So instead of you needing to take a blood samples, you can simply analyze the ISF that could be on site, and you can be able to diagnose this diseases based on that biomarker. Oral delivery, we haven't done much in the oral delivery. I would say we haven't done any yet in the oral delivery. There was a paper that was published, I guess, a year ago from MIT group, where they have developed a microneedle patch in a capsule. This capsule, once it's in the skin, start to dissolve, and then the needles would kind of fold out and press against the skin, the intestine, to deliver insulin. So like MIT have done quite a bit of research in this field. In terms of ocular delivery, we do have some work that is ongoing here for ocular delivery as well, using, in this case, of course, the hollow needles. And we have currently a graduate, PhD graduate student who is working on that. So now digging into specific examples, the first example is a drug. This is an oxytocin. It's a peptide hormone. It's used in the prophylactic and treatment of postpartum hemorrhage. And recently it has attracted attention to its peripheral actions in treatment of autism, schizophrenia, and in kind of like increasing the trust and helping with the anxiety. This is why they call it the love hormone. But apparently it's only available as injection. So again, it needs a cold chain of storage and transportation, and a need for healthcare provider to give the injections. And just reminding you that out of 25% of the maternal death in sub-Saharan Africa was due to the lack of the drug at site, basically due to these two reasons. So what could be better as a delivery system than the microneedle, which we developed in our lab? Here it was just a proof of concept, so we wanted to design a coated formulation, and these were on stainless steel microneedles. And for this project, we had set certain criteria that we wanted to meet. First, we want to be able to load the target dose. So we had to screen different formulations, different vehicles, different concentrations to meet this target dose of 20 international, around 10 international units, or that was around 16.7 or approximately 20 microgram per array of 5 needles. And that was achieved here with a concentration of 10% in ethanol solution. Of course, this has developed into a more sophisticated formulation later, as you would see. The second thing is we wanted to be thermostable. So we wanted to avoid the cold chain. So here we developed different formulations, and then we put them at stability at 40 Celsius and 75% humidity. And the reason for the humidity is this is actually mimicking the scenario where these needles would be used. It's tropical conditions, high temperature, and high humidity as well. So here, as you can see, these formulations that I'm pointing out were very bad in stability. You can see loss of the activity as soon as 1 week. Whereas here in this formulation, and the only difference in this formulation is that it was stored dry, meaning it was only subjected to the temperature effect, but they were stored with a desiccant. So they were not exposed to humidity. And that was actually even better than the pitocin, which is the injectable form of the oxytocin that was stored at 40 Celsius. So that has made us wonder that oxytocin is not, it can maybe with a certain formulation withstand the effect of temperature, but certainly not humidity. And that made us think, how can we stabilize it at humid conditions? Because this is, again, the situation that it will be used at. And just we screened a lot of formulation, a lot of excipients, and we ended up having a very good data with polyacrylic acid. We were able to demonstrate 100% of the activity retained after 2 months at 40 C and 75% compared to a control that didn't have any of the polyacrylic acid. So that was really a good finding, an interesting finding as well. Now there's 2 things that we want to make sure of, that we can attain fast skin delivery and we can attain also efficient skin delivery. Because imagine if it's something used as a life-saving medication in case of postpartum hemorrhage. We want to make sure that the drug is being delivered very fast. So here, these are a microneedle patch with a PEA, with the polyacrylic acid, that have been worn at different skin times. So basically applying the patch and leaving it for either 1, 3, 5, and 10. And then we would determine the amount of the drug either deposited on the skin or the needles. And from that, we can determine the amount of the drug that was delivered into the skin. And this is what's represented here by the dashed bars. And of course, we know how much we have initially, and we can compute from that the delivered amount. And as you can see here, comparing the dashed column, that basically we were able to deliver almost 80% of the drug, and it didn't really matter how long we leave it into the skin. So as fast as 1 minute, we can deliver quite a portion of the dose that was loaded onto the microneedle patch. Then moving into the vaccines, of course, everybody knows that the vaccine is a part of the being prepared for a pandemic, that its availability is really a critical part of being able to do vaccination, but it's not that only. There are associated things that we need to take care of and plan it, of course, and account for the expenses that these will have. One thing is the availability of the healthcare workers that will give these vaccines. They are mostly in the form of injections, so we have to have them available all the time to give the correct dose, to reconstitute the vaccine, and to make sure that the needles are disposed properly. And of course, having a safe disposal system implemented, again, to prevent any risk injuries, and of course, a cold chain of transportation and storage that cannot be disrupted at any time. All these, like we're aware of, all these like kind of were a reason for the delay of having mass vaccination, for instance, in case of the COVID vaccine. Yes, the vaccine was available in a relatively short time, but all these associated things and the need of a cold chain have definitely affected being able to vaccinate a quite amount of people. And that, of course, put a risk of having mutation into a virulent or more malignant or more resistant variants of the vaccine if we were not able to get the vaccines to as many people as we can at the same time. In terms of the studies that have been done on vaccines so far, there are quite many vaccines that have been tested for delivery using microneedle patches. Here are just some of them grouped into this table, and you can see that they vary according to their type. So we have here nucleic acids, VLPs, have the inactivated viral vectors, and soon we'll have the mRNAs as well. Here in this talk, I'm going to just focus on some of these, the Ebola, influenza, measles, polio, and rubella. So speaking about the influenza, just throwing some highlight on our human study that was published 2017 in the Lancet, and I'm here quoting that this is the first in-man study that shows that the use of a single dissolvable microneedle patch for influenza vaccination was well correlated, resulted in a robust antibody response, and was preferred over conventional influenza vaccination with needles and syringes. So this was really interesting findings. In this study, we had 4 arms. The first is the IM group that was given by a healthcare worker, which is represented here with this blue bar. And then followed by this pink bar is the microneedle patch that was again applied with a healthcare worker. And then the green bar here is a microneedle patch that was self-administered, which was something that we wanted to test the feasibility of the patient himself being able to apply the microneedle patch to himself. And finally, it was a placebo group, which is the purple column at the end here. And as you can see here with the seroconversions, these 3 groups were basically the same. Here it's maybe less for the B strain, but I mean, comparing a microneedle patch that have been inserted by a healthcare worker or the patient resulted in approximately the same immune response. And this was very interesting. And patients were asked if they would prefer a microneedle patch over an injection, and their feedback was really good, that they would prefer definitely a microneedle patch over a needles. And in terms of side effects that we have seen through this study, were basically very simple, like something like redness or pruritus that subsides with time. Another project here that was the Ebola microneedle patches, and in collaboration with Emory, we were able to formulate this into coated microneedle patches with or without an adjuvant that they have, and they wanted to test its efficacy. And as you can see here with the microneedle group, we were able to see a better antibody response. And with the adjuvanted groups, both responses were high, and they were basically equally effective. In terms of the photoprotective efficacy, both adjuvanted groups were able to show 100% survival, whereas the control or the IM group showed only 20% survival. So basically only 1 mice was able to survive the challenge. So it looks like we were able to coat a stable vaccine onto the microneedle patch, and it was able to induce a stronger antibody response and again protect against a lethal dose that was given to the vaccinated mice later. Another project that we were working on in our group is an IPV microneedle patch. And here I'm showing only the immune data. So here, these are two groups, an IM group and a microneedle patch group. And here also I'm showing only type 3. Usually IPV has two strains, type 1, 2, and 3, but I'm not showing type 1 and 2 here because it was, easy to work with in terms of stability and a proper immune expected response, where with type 3, it was a bit problematic. As you can see here, the immune response generated from the microneedle patch for type 3 was not as good as the IM. So we thought that this could be because the formulation that we are making is affecting how the vaccine is. So in this case, what we did here was took a patch, reconstituted it, and then injected into the animals to see if that hypothesis is true or not. But actually, it was not. And as you can see, the immune response to the injected reconstituted patch was okay. We wanted to boost the animals and see how that looks like. And actually, once we boosted the animals, all the groups looked the same. So it looks like something is still happening here that we don't understand, but once they are boosted, they are all have the same immune response. So we thought, okay, so maybe it's a dose thing. If we have a higher dose in the microneedle patch, would it be comparable to an IM group at this case? So here we designed this study where we have only one IM and two microneedle patch groups. The microneedle A group would have a 300% dose, and a B would have a 700% dose. These are divided among 3 doses. So after the first dose, B group showed a little bit higher, which is cannot be, I would say, explained at this point because it's basically the same dose as the A group. With increasing the second dose and the third dose is when we see a better response for the microneedle patch group. So it looks like with type 3, there's more things that we need to understand. And we did a lot of studies to kind of understand why we don't see a better immune response with type 3. And that would be a publication soon. Moving to the MR, or the measles and rubella microneedle patch, which is what we are working on currently in our lab in collaboration with the Gates Foundation, CDC, and MIT as well. On the left side here, you see a full microneedle patch that is able to load a full human dose of 10 to the power 3 of both measles and rubella, and these are presented here by these two bars. Basically, these are just the stock, and these are the concentration, and this is just the effect of loading less drug onto the microneedle patch. In terms of stability, we were able to show good stability data at 5C. However, with 40C, we were still working on optimizing the formulation. As you can see clearly here with the measles, we start to see a drop in the activity. Same thing here with rubella. In terms of immune response, patches are working fine. We were able to demonstrate a strong immune response in cotton rats, which is the model used here for measles, comparing a microneedle patch to a stratum corneum, and for rubella, comparing again a microneedle patch to a stratum corneum. But here you will see that we needed to boost the animals in order to get a better immune response in guinea pig. In rhesus macaques, we were able also to show a good immune response. However, here we didn't have a control group like a subcutaneous group because we had only 4 animals and we wanted only to test them for immune response. And that was all exciting in the sense that we were able to start our clinical trial that is currently going on in Gambia. It's a double-blinded, randomized, controlled phase clinical trial. It's the first clinical trial of microneedle patch to test children which is something that we're very excited about. And we will be able to get some data next year, early next year. In terms of future perspectives here, I'm going to be really quick. I know that actually time is up. What are we actually doing right now at Georgia Tech? So we are having collaboration with MIT, and we're working on what we call an on-patient medical record. So basically delivering near-infrared particles that once delivered into the skin would leave something as a pattern. And this pattern could be something like a code, as you can see here, that is connected to a database on the cloud that once the patient or whoever wants to detect the vaccination status can just use a cell phone and just scan that to kind of determine the vaccination time, the vaccination type, and all basic information about the time of the vaccination and where it was done. So this is something that we're very interested to pursue with MIT. The other thing, of course, is the mRNA vaccines. We're very excited because right now there are research groups that have started already working on delivering of COVID spike protein using microneedle patches, and they found really good results. Another results that was really interesting to us is an intradermal dose that resulted in a dose sparing. And this is very important because imagine having a limited supply of COVID vaccine where you can maybe vaccinate a large number of people with the same amount of stockpile. So, and of course, we cannot load much in a microneedle patch. So this is something that we are very excited to pursue, again, in collaboration with MIT and other groups as well. The improved stability and the rest reliance on cold chain is something that we need to still work on, because you know that mRNA it's not only mRNA, it's the lipid nanoparticles that you need to make sure that once they are dried into a microneedle patch and reconstituted in skin, they reconstitute in a form that can still maintain the protective efficiency for the mRNA and being able to transfer it to the cell. And it's not only us, it's the government and the BARDA that is also getting into the sense that microneedles are very important in the vaccination, and by making large amount of funds available for other groups working on the delivery of SARS-CoV-2 spike protein microneedle patches. So what are the obstacles or the uncertainties? There are still uncertainties about the safety of the microneedle patch, but so far, all the clinical trials turned out fine. There are no red flags. Things are going well. The large-scale manufacturing is still an issue. We expect it to be cost-effective and reliable, but as many products hits the market and we can learn more about how we can make this better. Sterility requirements, we are still not sure if it's like a small bioburden would be good, or does the patch has to be sterilized? You know that aseptic manufacturing would be very expensive, and that would add a cost to the microneedle patch, and we want it to be affordable. And also the thermosterilization is not possible with active ingredients that might destroy it. So is it a low bioburden is sufficient or not, since it's just a breach of the sterile corneum? But there are evidences in our life that actually say that it might be okay to use just a low bioburden. Like if you are aware of the derma rollers that you can buy off Amazon, you just roll it on your skin to kind of help with the wrinkles and the scars. And these are not a single use. You can keep using it without the need to sterilize it in between uses. And there haven't been any reports that there was any issues with that or any safety concerns. Of course, the other concerns would be being able to manufacture a patch that has an accurate dose, being able to deliver large doses. Again, we are confined with a small volume, but we can mitigate this by making longer needles, larger patches, and this is what we are working on. Possibility also of minimizing skin irritation with using excipients that wouldn't provoke this skin immune response. And also the other factors that we cannot control, it's out of our hand, that might affect how the microneedle patch behave once inserted in the skin. So in conclusion, dissolving microneedle patches is definitely better than a hypodermic injection if we are thinking about a vaccination campaign. And that when applied to specifically COVID, it has a great potential to improve the vaccination coverage and save many lives. And while we're not ready right now with a microneedle patch for COVID, hopefully we can be ready for the sad to say, but hopefully we can be ready for the next pandemic with an implemented infrastructure of manufacturing and regulation. With that, I would like to thank my group, Dr. Mark Prosnitz, and all my colleagues, and also the funding sources for our projects, Bill and Melinda Gates, NIH, and from others. And with that, I would be happy to take any question. Thank you so much.